Hi again, this is Dr. Goldsmith. This is the third in the series of videos that I'm leaving for you in my absence. We now have mature but naive B and T cells based upon what was just previously described. So what's going to happen to them? How are they going to be activated, and what do they do once they're activated? So first I'm going to look at T cell activation, and then next time we're together we'll move on to what the effects of those T cells are. So I'm going to give you some background, quick refreshers, and then talk about the phases of the T cell response. T cells, as you I'm sure recall, mediate cell mediated immunity. In other words, they're part of the adaptive immune system. They're really interested in combating intracellular microbes. The microbes might be things that have evaded phagocytes, microbial or microbicidal activities. They could be found in the vesicular compartment. They can proliferate in the cytoplasm. They may have come in from the extracellular environment, but they're associated with cells now. And they can infect a variety of cells via their receptors, and the cells are going to lack mechanisms, in this case, to destroy the microbes and proliferate in the cytoplasm. So, for example, a virus that, say, infects the liver, for example. The responses by the T cells are going to include many things. That includes the cytotoxic response by CD8 positive T cells. That includes helping macrophages with their activation. It also includes helping B cells produce antibodies better. And so common to all these functions is the fact that the T cell must interact with other cells, so phagocytes or B cells and so on. So they're cell-mediated immunity. The T cells are going to specifically recognize peptides that are displayed by MHC molecules. You guys know this. They've been processed. They're peptides only. And it ensures that the T cells are going to see and respond only to antigens associated with other cells. So over the next few minutes, I'm going to focus on the signals that are needed to activate T cells, the receptors that sense and respond to these signals, how we can have only a few mature, naive T cells that are specific for a microbe that are converted to the large numbers of effector cells that we need. Obviously, there's lots of proliferation and differentiation that goes on. And then I'll speak briefly, but not very much, on the molecules produced by the T cells that mediate communication with other cells. With regard to how T cells respond, the T cells have responses to cell-associated microbes. As you know, this occurs in certain phases. The first would be clonal expansion, where you get a series of steps resulting in increased numbers of antigen-specific T cells, and then conversion of these naive T cells to effector cells, so proliferation clonally, and then differentiation. And you need to think about how the T cells that are in circulation, they travel through the blood and the lymph, they continually circulate, they are mature, but if they're not exposed to their antigen, they're considered naive T cells. So they have to meet their antigen with MHC to participate in any sort of response. And if they do get that appropriate MHC plus peptide antigen stimulus, they will differentiate, and then they will become effector T cells. And I will do my best to distinguish between the two, whether we have a naive but mature T cell or whether we have an effector T cell. So this is just a graphic to indicate, you've got these T cells that circulate. This happens to be circulation through a lymph node, and we're having either a CD8 positive T cell, naive T cell responding to antigen presentation, or a CD4 positive T cell responding to its antigen plus MHC. They will proliferate, they will differentiate into effector T cells that will re-enter the circulation and migrate to their sites where they're needed and have their appropriate responses. In the case of CD4 helper cells, they will secrete cytokines that might help macrophages, for example, with their phagocytosis and killing. They might help B cells. That's actually going to happen somewhere else. And in the case of the CD8 positive T cell, they will migrate to the site of the infection and kill the infected cell. We're not really going to focus on the effector functions today. We're really going to focus on this stuff to begin with. So remember that adaptive immunity is initiated in the peripheral lymphoid organs, and naive T cells specific for an invader's antigen, which is going to be a peptide, must be induced to respond to infection by an antigen-presenting cell. Dendritic cells are the professional ones. They present antigen in the peripheral lymphoid organs like the spleen, the lymph nodes, the MALT, the BALT, and so on. The dendritic cells, recall, can pick up antigens at the site of infection and then mature as they travel to one of the peripheral lymph organs, or dendritic cells can pick up free antigen and travel through the lymph or blood to the lymphoid organs to uptake and present antigen, again, in the peripheral lymphoid organs. And again, recall that the naive T cells are going to be on the prowl. They're going to continuously circulate through the blood and lymph, passing through the lymphoid tissues along the way. They may encounter dendritic cells in the lymphoid tissues and sample the wares. We've seen that before. In other words, they're going to interact with the dendritic cell, sampling the peptide MHC complexes that the dendritic cell is showing on its surface. If that happens to be a match for the T cell's specific antigen MHC complex on that mature dendritic cell, migration of the T cell is going to stop and proliferation, that is clonal expansion and differentiation, will proceed. So that leads us to, how do the T cells that are mature but naive recognize their antigen, clonally expand, and differentiate? And it really depends upon antigen-presenting cell signals. So first I'm going to talk about antigen recognition, engagement of the T cell receptor with its specific peptide MHC complex, and then I'll talk about survival and expansion, and ultimately differentiation. So this is the big picture for those processes, antigen recognition being the first, MHC plus peptide being presented to a naive, mature T cell. If the T cell likes what it sees, the T cell receptor recognizes MHC plus antigen. The dendritic cell will supply certain signals that will include cytokines and will change cytokine expression on that naive mature T cell. It will start to proliferate, it will differentiate, and ultimately it will perform its effector function. So I'm really going to talk about the processes involved here today. So what about antigen recognition and co-stimulation? We'll start there first. I put this up early because we are going to be talking about many of these molecules that are listed on this slide, and this is a really good thing to come back to. Many of them you already know, which I'm sure you'll be thankful for. For example, this is the receptor that is seen, or all the receptors that are seen on a CD4 positive lymphocyte. These would be slightly different on a CD8 positive lymphocyte, but not extraordinarily different. And then on the antigen presenting cell, you see the ligands that are expressed on the antigen presenting cell. And you already know that a CD4 positive T cell has a co-receptor, CD4, that will bind to the MHC class II molecule. You already know that the CD4 positive, mature, naive CD4 positive T cell will express a fully functional T cell receptor that has undergone positive and negative selection and so on, as previously described. You already know that there are invariant signaling molecules that are associated with the process for getting signals transduced. The antigen receptor is all about diversity. These invariant signaling molecules are necessary for signal transduction. So you already know that there's CD3, you already know that there's zeta. So here they are, and they transduce signals. Haven't spoken about CD28, CTLA4, PD1 as far as signaling goes. And the final thing to think about is, how is that T cell going to bind and stay bound to the antigen presenting cell long enough for these processes to occur. That involves adhesion molecules, one of which is listed here, LFA1 on the CD4 positive lymphocyte, and ICAM is its ligand on the antigen presenting cell. Here's a listing of those molecules, their functions, and their ligands. Again, this is a really nice thing to come back to. By the time the next few segments of the videos are complete, you will have heard about all of these various molecules and their roles. So first up, I'm going to really focus on antigen recognition through the MHC complex and the associated peptides. We're not going to spend a lot of time on this since you have heard this over and over and over again. This is really a nice review, though. The T cell receptor recognizes antigen, and it's got CD4 or CD8 co-receptor, and these together are going to recognize peptide antigens and MHC on antigen presenting cells. This is going to initiate T cell activation. There will be a little more to it, which I'll talk about. Remember that any peptides that are processed from the vesicular system are going to be presented on MHC class 2 to a CD4 positive T cell, and any peptides coming from the cytoplasm will be processed appropriately and presented on MHC class 1 on the antigen presenting cell. Also recall that the CD8 positive T cells are going to recognize peptides that are presented by MHC class 1, and they will differentiate into cytotoxic effector T cells, or CTLs, while the CD4 positive T cells are going to recognize peptides presented by MHC class 2, and they differentiate into several different subtypes of effector cells. All of them help in some way. They might help macrophages, they might help B cells, or they might be involved in regulation. And then they have different subtype names like TH1, TH2, TH17, and we'll talk about those when we talk about the effects of the particular subtypes of CD4 positive T helper cells. But that won't be today. So in order to get your T cell activated, there are going to be biochemical signals for that activation, and these are going to be triggered by proteins that are linked to the T cell receptor and form the T cell receptor complex. And in addition, there will be signals generated by CD4 or CD8. So again, this is something you've heard before. The antigen receptors are diverse. They're all about binding peptide plus MHC appropriately, and they need a lot of diversity, particularly in their V regions. So they can recognize many microbes, but they can't transmit signals. We instead have these conserved invariant signaling molecules that are associated with the T cell receptor so that activation can occur. So antigen recognition and signaling are segregated into different sets of molecules. You have heard about this before. Now I'm going to expand upon what these molecules actually do. So recall that the T cell receptor is non-covalently associated with CD3, and CD3 is actually a 3-protein complex. In addition, the TCR, the T cell receptor, is non-covalently associated with a homodimer signaling protein, that is the zeta chain. So the T cell receptor complex includes the T cell receptor, CD3, and the zeta chain. That's the whole complex for antigen binding and for signaling. The T cell receptor alpha and beta chains, you heard how they are produced in previous recordings. They are variable. They recognize the antigen plus MHC. And then CD3, that 3-protein complex, and the zeta chain homodimer, these are conserved and they're necessary for signaling. Again, separate antigen binding and signaling, but they are non-covalently associated for the whole complex. Here they are visually, where you've got antigen recognition going on, your antigen presenting cell presenting a peptide on MHC class 2, and the T cell receptor will recognize that MHC antigen presented by MHC, and then its co-receptor will bind to the MHC molecule itself. We've got our 3-protein complex CD3 molecule, and we have our zeta homodimer. Both of these are involved in signaling. You'll notice they're showing signaling transduction here. And the CD4 T cell actually can be involved in signaling as well. One of the things to note on these signaling molecules, there are particular motifs that are associated with them. You've actually seen these before. These are immunotyrosine motifs, and they are for positive signaling, so you'll see them both on the zeta chains and the CD3 chains. So you'll get signaling through them. There are some other important molecules involved, as noted on that sort of summary slide before. Naive T cells, they're on the prowl. They're going to bind transiently for a short period of time, to antigen-presenting cells, and they're going to sample the MHC molecules plus the specific peptide that the antigen-presenting cell is showing. If they encounter their correct specific complex, the T cell signaling complex induces a conformational change in an adhesion molecule, LFA1, which is on the T cell. That is through signaling through those associated invariant signaling molecules. So one of the outcomes is to make a conformational change in this adhesion molecule, LFA1. The conformational change increases LFA1's affinity for its ligand, which is ICAM1. This is found on the antigen-presenting cell, and tight binding will ensue. This association of the naive but mature T cell plus the dendritic cell can go on for days. And they take it to that dark corner in the bar and they hang out together for a while. More signaling occurs and we get proliferation as a result. Proliferation of the naive T cell occurs. The progeny from this T cell, so the daughter clones of that original T cell, are also going to bind the antigen presenting cell, and they will differentiate into effector cells. So the antigen presenting cells are going to be there and provide signals for clonal expansion and for differentiation. And that's illustrated here, where we have a T cell and it's got its integrin, the LFA1, in a low affinity state while it's mature but naive. It samples the wares on an antigen presenting cell. If it finds an MHC molecule on the antigen presenting cell plus peptide that it likes, signals will be sent through those invariant signaling molecules. The signals will provide a change in the affinity of that integrin, the LFA1, so it becomes a high affinity state integrin, illustrated here, and it will bind its ligand, which is ICAM1, on the antigen presenting cell, and you'll get that association to go on for days. This should really sound familiar. It's very, very similar to having signaling and association of the cells for longer periods of time and changes of affinity, because you heard about it with leukocyte extravasation. I'm going to stop here, and then I will move on in the next video for survival and clonal expansion.